Hello, I'm James Edwards, and welcome to my presentation on a review of recent evidence for curcumin as a potential therapeutic agent for Alzheimer's, Parkinson's, and Huntington's disease. Introduction. Neurodegenerative diseases such as Alzheimer's, Parkinson's, and Huntington's disease can cause significant pathological neuronal loss and cognitive and physical decline in up to 50 million people worldwide. The Alzheimer's Association recently estimated that there will be a new diagnosis of Alzheimer's every 33 seconds by 2050 due to an aging population worldwide. This is one of the main reasons behind a demand in new research into new treatments for neurodegenerative diseases to treat both the symptoms of these chronic diseases as well as slowing or reversing neurodegenerative pathologies. Curcumin is an active hydrophobic phenolic compound which has remarkable pharmacological properties. It is both an antioxidant and an anti-inflammatory, and they therefore may have the potential to be an effective therapeutic agent for neurodegenerative diseases. The focus of this narrative review is primarily on testing curcumin as a potential therapy for neurodegenerative diseases in human clinical trials. However, animal and cell studies have been included where there have been no human trials. Methods. Two databases were used as the site for our searches, PubMed and clinicaltrials.gov. This helped us to identify relevant studies and trials which would match or could be associated with our search terms as seen in the table. Statistical analysis was not completed in this narrative review due to a wide variety of different data collected as well as the small number of studies and trials included. Results. Alzheimer's disease. 9 human clinical trials were identified for Alzheimer's. 6 of these studies demonstrated an improvement in cognitive function, inflammatory, and pathological markers. When comparing trials using just healthy adults, we can see that 75% of trials found a positive impact. Alternatively, trials using participants with diagnosed Alzheimer's or mild cognitive impairment found that there was a positive benefit in 60% of trials. All 3 studies that lasted 4 weeks long or less found there to be a positive impact on healthy volunteers. No short-term study has been completed on patients with Alzheimer's. 40% of long-term studies showed an improvement following curcumin supplementation. These studies found improved memory and cognitive ability in patients. Results. Parkinson's disease. No human clinical trials have been completed on Parkinson's. Therefore, we have used 4 animal studies and 3 cell studies. There was an effectiveness of curcumin in all of these models. Some of the positive benefits are listed here. Results. Huntington's disease. No human clinical trials have been completed on Huntington's disease. Therefore, we have used data collected from 5 animal studies. There was an effectiveness of curcumin in all of these models. Some of the positive benefits of these models are shown here. Conclusions. Further studies are required for both Alzheimer's, Parkinson's, and Huntington's disease, despite 86% of trials demonstrating an improvement in cognitive function, pathological, or inflammatory markers. Further studies could also look into improving the bioavailability of curcumin via nanocurcumin particles. This could potentially help to improve the effectiveness of curcumin as more may be able to cross the blood-brain barrier.